Hello students, hope you are enjoying study at your home. In the previous video, we discussed about the structure of ovule. Now, let's take the more details about the structure of female gametophyte. In the previous video, I told you that inside the ovule, the female gametophyte, which is embryo sac, is present. Embryo sac is present inside the ovule, and this embryo sac is also known as female gametophyte. Now we will discuss about the structure of female gametophyte. This female gametophyte is present inside the ovule. This is the normal structure of ovule. This type of ovule is known as anatropous ovule. In the case of most of the families of angiosperms, the ovules are the anatropous ovules, which are known as upside down ovules. This is the anatropous ovules. Besides this, ovules have different type of structures and shapes. We will discuss in the later part of the chapter. But now you have to keep remember that type of the ovule which are present in most of the families of the angiosperms are the anatropous ovule. This anatropous ovule this is the same thing we discussed. This is the funicle, hilum, the point where the ovules joins with the funicle, and this is the complete structure of the ovule. Inside the ovule, the tissue is present, that is nucellus. Nucellus is present inside these integuments. Nucellus is present. Now let's see how the female gametophyte develop from this nucellus. When female gametophyte have to develop, out of this nucellus, it has many cells, means like many cells are present here, and it is rich in cytoplasm and abundant of cells are present here. Many cells are present here in the nucellus. For the formation of female gametophyte, one cell of the nucellus grow into the size. एक cell जो होगी वो size में increase हो जाती है. In the case of when female gametophyte की development होगी, nucellus से एक cell जो है अपना size increase कर लेती है, and that cell should be present at the micropylar end. Like in the previous video, I told you when we talk about the female gametophyte, we have to always mention the pole. So this is our micropylar end and this pole is our chalicer end. Micropylar end पे एक cell होगी वो size में increase हो जाएगी, and that cell is known as MMC. This cell is known as MMC. MMC means Megaspore mother cell. This is MMC is Megaspore mother cell. This MMC will increase in the size and it will now start dividing. Here one more important point should be keep remember here, that nucellus has diploid in nature. ये जो nucellus होता है, इसकी जो nature होती है diploid होती है. Means all the cell of nucellus are diploid or 2n in numbers. When one cell grow from this nucellus MMC, that is also diploid. Now let's see how it will develop into the female gametophyte. This MMC, this cell which is 2n or diploid in nature, it will start dividing. It undergoes through the meiotic division. It undergoes through the meiotic division. So meiosis will happen here, and due to the meiosis, 4 cells are formed here, and all these 4 cells are haploid, and these 4 cells are arranged together and they form a tetrad. This is known as Megaspore Tetra. Megaspore tetrad. Like in the case of microsporogenesis, also we talk about the tetrad, where the tetrad had the different shapes. In the case of Megaspore tetrad, there is only one shape, that is linear shape. So in the case of megaspores formation, Megaspore tetrad is always in linear shape. So all these cells of these tetrads are haploid in nature. These all cells are haploid in nature. Now let's see what happens further. When these tetrads are formed, out of these tetrads, these all 4 cells do not take part in the further development. सभी की सभी चारों सेल आगे पार्टिसिपेट नहीं करती. Out of these 3 cells get degenerate. Out of these tetrad, 3 cells get degenerate and only one will survive. The cell which is present, if we mention the ants here, this is the chalazel ant and this is the micropylar ant. This three cells of the tetrad which are towards the chalazel ants and the one cell which is towards the micropylar ant, these three cells get degenerate. So they get degenerate. अब हम देखते हैं कैसे, अगर इसको दिखाना है तो हम ऐसे शो कर सकते हैं. These are the three cells which are degenerating. And this is the one cell which is towards the micropylar ant. This is the one cell which is towards the micropylar ant. It will survive. These three cells get degenerate. Only one megaspore will take part for the further development of female gametophyte. That type of development of female gametophyte is known as monosporic development, because only one megaspore will take part in the female gametophyte development. Now what happened with this one cell, this one megaspore? It is N in nature. It's haploid in nature. Due to the meiosis here, the haploidy is changed from 2 and all these cells are become haploid in nature, and this haploid cells will develop further for the development of female gametophyte. In the development of female gametophyte, it's so that out of 4 cells of the megaspores, out 3 which are towards the chalazel and get degenerate, and only 1 remain functional. So this is our functional megaspore. We can write here, this is our functional megaspore. Now what happened? This megaspore, this functional megaspores now do the three successive meiotic division. यहाँ पे अब इसके अंदर माइटोटिक डिवीजन, यहाँ पे माइटोसिस होगा एंड जो माइटोसिस होगा ये लगातार होगा, एक के बाद एक 3 माइटोसिस डिवीजन इसके अंदर होते रहते हैं. बट यहाँ पे एक इंपॉर्टेंट चीज जो आपको याद रखने वाली है वो ये कि जब इस फंक्शनल मेगास्पोर में माइटोसिस होता है, ये एक इस तरीके का माइटोसिस होगा जिसके अंदर ओनली कैरियोकाइनेसिस विल टेक प्लेस. Only karyokinesis will take place and the cytokinesis will take place later on. So let's see what happens. The nucleus of this functional megaspores get divided due to the first mitosis. This is the first mitosis. First, due to the first mitosis, it forms 2 nucleus. Only karyokinesis will take place. Cytokinesis will not take place here. So one nucleus will divide into the 2 nucleus. Then second mitosis, these 2 nucleus will form 2 more nucleus. This is the second mitosis. Later on, it will divide through the third mitotic division, and in the third mitotic division, these nucleus will form another nucleus. And finally, we got 8 nucleate structure. These are the 8 nucleate structure after the third mitosis division. Karyokinesis is only take place here. Cytokinesis will now it will take place. But let's see what happens when this structure is formed. It is now 8 nucleate structure in which the 8 nucleuses are present. So it is known as 8 nucleate structure. Now let's see. After the formation of 8 nucleate structure, the cell wall formation will start making, and these nucleus will arrange in a proper manner. Out of these 8 nucleus, the 3 nucleus will come together and they form a cells. They form the cells. They form the cells. The 3 nucleus which will arrange towards the chalazel and 3 nucleus. Let's mention the ant here. This is our chalazel ant. We are mentioning here from starting chalazel ant, micropylar ant. This is also chalazel ant and micropylar ant. The 3 cells or 3 nucleus will arrange towards the chalazel ant and they form cell wall towards and become the 3 cells. These 3 cells are known as antipodal cells. These are the 3 cells which arrange towards the chalazel ants are known as antipodal cells. Antipodal cells. Rest all out of these, 3 more nucleus will arrange themselves towards the another ant, that is the micropylar ant. So 3 cells will arrange, 3 nucleus will come there and they arrange in the form of 3 cells, and these are known as, these two cells are known as synergids, and this one big central larger cell is known as egg cell. The 2 side cells are known as synergids and one central larger cell is known as egg cells. These synergid and egg cells combinedly known as egg apparatus. Combinedly we can use term here is egg apparatus. Out of 8, 6 nucleus, 3 towards the chalicer and 3 arranged towards the micropylar end, and 2 nucleus which remains, they form a large central cells in the center of this embryo sac. So in this stage we can say here that in out of these 8 nucleus how they arrange. Let's see one more time. यहाँ तक हमने देखा due to the 3 mitotic divisions, one megaspore nucleus is divided into the 8 nucleus. एक megaspore का nucleus था, उन्होंने 8 nucleus form किए, and now these 8 nucleus arrange into a different manner. 3 nucleus they move towards the chalicer ant and they form the walls around it and form the 3 cells, and these 3 cells which are present toward the chalicer ants are known as antipodal cells. This 3 cells which move towards the micropylar ant, they are arranged here in different manner. One cell will become larger and there 2 cells are also present there. This combination is known as egg apparatus. Out of this one larger cell, जो बड़ी cell है उसे हम बोलते egg, that is the female gamete, and with the egg 2 cells are also present. These 2 cells are known as synergids. The complete combination of synergids and egg is known as egg apparatus. And rest 2 nucleus, जो बच गए हैं but 2 nucleus, they arrange themselves in the center of the embryo sac by forming a single larger central cell. This is our central cell in which 2 nucleus. This is our central cell, and in this we can see all these cells are N in nature because they have only one nucleus, so they have haploid in nature. Antipodal cells are haploid in nature, egg is haploid in nature, and these synergids are also haploid in nature. But this central cell which contains 2 nucleus, we can say that the ploidy of this cell is 2N. It's diploid in nature. In this way, we can say in the embryo sac, the embryo sac is formed in such a way in which we can found 8 nucleus and 7 cells. 3 cells towards the chalicer end, 3 cells towards the micropylar end, and 1 cell which is present in the center. This type of arrangement of embryo sac is known as 8 nucleated, 7 celled embryo sac. So it's 8 nucleated, 7 celled nucleate. It's 8 nucleated, 7 celled arrangement of embryo sac. This is the characteristic feature of angiospermic embryo sac. Here one more thing we should keep remember, with these egg apparatus in the egg apparatus, these synergid cells have some finger-like projection. They have some finger-like structures. These finger-like structures are known as filiform apparatus or filiform cell, because these are the structures which guide the pollen tube during the process of fertilization. When fertilization takes place, during the fertilization after pollination, when pollen tubes form, pollen tube come inside the ovary and it moves inside the embryo sac through the micropylar end. At that time, to guide the pollen tube so that it can enter inside this ovule, to guide the pollen tube, these filiform apparatus helps or guides the pollen tube for the entry of this pollen tube inside this embryo sac so that it can get fused with the female gametophyte. This is the structure of female gametophyte, in which egg is representing here as a female gametophyte, and after that we will discuss about the process of pollination, in which the pollen grains reaches to this part, this female reproductive part, so that it can be easily get fused with female gametes, that is egg, so that fertilization process can take place. Thank you. Keep watching and keep studying.